I am ready to celebrate with your presentation or connection with your research activity.
Yeah,
so
well,
you have done maybe more than 40 years already, no? If you
that's
start
right. No, yeah,
huh?
I came to the US 1990.
1990, yeah. And you started before in Berlin.
Yeah, yes, I spent 11 years in Berlin.
Yes. Yes. Yeah. It is the calcium, what you call it, calcium receptor
Binding.
protein. It's also protein in the membrane.
Calcium binding protein, yes,
Binding
that was
protein,
the,
yes.
you know, it's,
and also the transport.
That's right. Well, it's now used for as a marker of cell death.
Oh,
and
marker of cell death.
Yeah, when the cell died, so this molecule also spilled, you know. Yeah.
Okay. You need to close to stop first your share.
Yeah. Okay.
Yeah, I want to
yeah.
Okay.
That's fine. Seems like it worked.
Just want to. Yeah, I just want to explain shortly to the participant about your CV.
Sure. I will try to speak Indonesian, but I don't know how. Maybe
how many I think we have more than 100 participants. More than 100 participants, and maybe one or two from Singapore.
Okay.
I don't know exactly, but yeah, you can mix, of course, yeah.
Okay. I see Dr. Anton. Good morning.
Yeah.
Good morning, Professor Lim.
Dr. Anton.
Nice to see you again.
Nice to see you. So.
Thank you for your kind contribution.
Well, I hopefully useful. Yeah.
I would like to say that happy birthday to Professor Chris.
Yes. Let's hope this is a good, a day to start.
Yeah, that means that it's not the final one. We need to have also another presentation from you, because I think especially for the student, the lecture about fundamental of inflammation is very necessary.
Yeah, well, it's very true.
It is connected with many diseases.
Yeah, I think nothing, all the processes involve inflammation. Almost,
Yeah. Okay, we arrange.
Oh, that's an old picture.
Yeah. Old picture. No,
it was done by people at the Kick Andy. That was
Kick Andy. Yeah.
And they have a photograph.
But it seems to me that there is no change after years.
Sorry, no change.
Yes, no change.
Oh no.
Selamat pagi, Dr. Mardi.
Ya, Dr. Mardi.
Dr. Mardi is here too. Huh?
Ya.
Dr. Mardi masih di mute.
Ya, Dr.
Hello, Prof. Lim. How are you?
Oh, fine, thank you. How are you?
long time.
Yeah, maybe, yeah, couple,
5 years.
5 years
Yeah.
at least, yeah.
Yeah, you never back to Kelapa Gading.
Yeah, with the COVID now, no chance.
Yeah. Now danger. Kelapa Gading is a red zone already.
Yeah, yeah. I
Red
think
zone.
Sunter, right? Sunter.
Yeah, Sunter also.
Yeah.
Mostly Jakarta is red zone.
Yeah, yeah. But,
Especially for the in the urban area.
Yeah. So it's interesting here. I keep reading stuff. So and I think it's possible that the intriguing part is children can carry this without any symptom whatsoever. And that maybe is the culprit or the problem. They can spread the virus easily because nobody knows, and so one of the report is from Boston. They found high titer of the virus in children that do not show anything, much higher than people, elderly who's dying in the ICU. The titer of the virus. So it's amazing how this
have a high heart immunity.
Yeah, well, we don't know what caused it. Maybe it's the T cells or something. It's interesting.
Yeah, we did to start our seminar. Yeah, so I have already introduced your CV in a slide forms, so that I think I don't need to repeat it again. But first of all, I would like to thank for your willingness to share your idea, your research results to all of us. We hope that we don't want just to learn your presentation, but also to learn from your spirit to do research activity for I think more than 30 years already. Yes, starting as you were in Berlin as a student at that time.
That's right.
So please, Roflim, you can start with your lecture. And,
okay.
Yeah.
I will try to share. Okay. You can see the screen?
Yes.
Okay. All right. So what I'm trying to do today is just to, tell you the story of my experience doing research, especially to develop a drug that can be useful. So I don't know. Should I speak in English? Maybe it's okay, or switch to Indonesian.
I think in English is okay because maybe there are some participants from other country.
Okay, I will try to speak slowly and clearly. Hopefully, you can understand me, follow me. So the title is, basically the idea is this protein that I have been working maybe 20 years. And only in the past 5 years, it became clear that this protein is very, very intriguing and very complex and very interesting, put it that way. So let me start with, the idea of inflammation. I used this to introduce it because Prof. Chris just 2 days ago mentioned that maybe also students will participate in this. So I just want to make sure that, so inflammation is a good thing in general. So it's a physiological process, a necessary defense mechanism of the body when we have a bacterial or viral infection, and it helps us to heal the injury. So as we know, in the medical school especially, so the 4 signs is redness, heat, swelling, and loss function. That's a typical inflammatory reaction that we can see or feel. So, but it become a problem if we're talking about systemic inflammation, because it's a syndrome that the whole body inflamed. So, and this is really a challenging situation that 18 million people worldwide suffer annually. And in the US alone, it's like 700,000 new cases every year. And the mortality rate didn't change in the past 10 years, 30 to 50%. And this is in the ICU. That means that with all the advanced support, as you can see in the picture on the right, a lot of support. This is a burn patient that get all the support from the kidney, the lung, and so on. Still 50% died. So it is a leading cause of death in the ICU. One of the problem is there is no ideal marker. Currently only blood culture is available to confirm this, and usually it's not very, very sensitive or not late in the game, because if you find a bacteria in the culture, it takes already couple days and so on. So it's usually late. So, and more troublesome is there are at least 30 drugs tested in clinical trial for sepsis, and all failed in the past 30 years. So it really become skeptical for people to study this. And so, actually some pharmaceutical company just give up. They don't want it to be involved or invest in this anymore. So, and again, there is no FDA approved drug currently available that specifically to treat sepsis as a specific drug. So with that, so that's the information. So why is it so complex? Because there is at least two forces that come. So one is the microbial agent itself. So that can be virus, can be bacteria that basically secrete or release this called PAMs, pathogen-associated molecular patterns. And that's cause basically the body to react with cytokines and nitric oxide and so on. That is actually the start of the inflammatory reaction. And if you hear about cytokine storm with the COVID-19, that's part of that, because the virus created this or triggered this reaction of the body, and that caused this systemic inflammatory response syndrome or sepsis that can continue to cause a multi-organ dysfunction and finally septic shock. And when it happened, a lot of cells died, and the tissue damage causing cells spill over the content, and some of that content we call it DAMs. So it's abbreviation of damage associated molecular patterns. That could be like a histone, a heat shock protein, and so on. It's something that may be in the nucleus that spill over, and when it goes to the system, then the immune cell react and then secrete cytokine and so on. So this cycle is the problem, and this involves so many pathways. I just put here just to show how complex it is. It can involve complement, coagulation, endothelial cells, epithelial cells, and everything else, and then the blood and lymphatic cells that all secreted mediators. And that is what, sometimes it's overwhelming to the body and sometimes referred to cytokine storm. So, and that will affect the brain, the lung, the cardiovascular system, kidney, liver, gut, and microcirculation. So this is the problem of this sepsis. And so it's without, when I started the research, I wasn't even aware of this problem. If I knew, I may not wanted to be involved in this, but so it was just accident that I end up to be involved in this field, very, very challenging field. So as you can see here is the 2D gel electrophoresis of a plasma. So if we let a blood sit and the plasma, we separate it in the gel, there's at least couple hundred or thousands of protein. Every single spot represent a protein. So, and that protein actually is very important. So one of those is we call inter-alpha inhibitor proteins that I accidentally actually discovered or working on. So, and it turned out after many years. So this is the summary here. It turned out so the protein is a key element of innate immunity. So involved in the host response. So it modulates a lot of things. So one of the first thing is proteases or enzyme. So it inhibits proteases. So that's the why we call the name inter-alpha inhibitor, because of initially, it called inter-alpha trypsin inhibitor. So because the first time it was discovered, it inhibit trypsin. So now we know it inhibits many serine proteases. So, and inter-alpha refer to the mobilization or the gel electrophoresis. So because there is an alpha, there is a beta, and there's a gamma globulin in the proteins. So, and this protein run in the electrophoresis between the alpha chain. So basically they called inter-alpha. So it not only inhibit proteases, but it block complement, downregulate pro-inflammatory cytokines, binding danger signals. That's, I will go through that. And regulating cell morphology. This is very interesting, especially the neutrophils, and protecting the endothelial cells. So this all, what I list, this is important in the process of sepsis actually, or systemic inflammation. So what we early on found out is the level of this protein is high, but depleted, or we lost, or because it's consumed, so it lost the majority of this protein, so it depleted during systemic inflammation. So, and we thought if we basically normalize the level, maybe we can somehow correct the problem, and it turned out it may be true, so that we have data supporting that. So what we believe is inter-alpha inhibitor therapy may be a unique approach to provide broad immunomodulatory effects in severe diseases. So in contrast to targeting only single pathways, so many drugs that in the past developed is targeting very specific to one problem. Either it is a TNF alpha or interleukin 6. So very specific, but then mostly failed because the problem is not just a single problem; it's a many problem. So, and we hope that that inter-alpha inhibitor can offer a better approach. So because of the structure of this protein that is so complex. So I will go through that. So, but initially, so I mentioned this protein is not, it's known already in 1910. So it called initially inter-alpha trypsin inhibitor because it inhibit trypsin initially, and they found in the 50s that is actually excreted in urine. So you can find in urine this protein and still active. It's very, very, very unique actually. So, and then not until 1970, people can link it in the serum and isolate it from the serum. And then there's a study in the structure and only in 1990s, end of around 1990s, the anti-inflammatory function of this protein become a little bit clear. But so, and then in the 2000s, so there is a knockout mice of this protein. So, and that's even, give evidence that this molecule is maybe important. So until today, there is no human detected without this gene of inter-alpha inhibitor. So it's maybe lethal without, inter-alpha inhibitor. So there is no human detected with this, lacking this gene. So, and so anything in this colored labeling here, so this is our contribution. So so my group research that, so in the past, basically 10 years, we work very hard to understand this. So initially in sepsis, anthrax, and then later on we found that it's expressed in the brain, which is interesting, and then with the danger signal neutralization and so on. So I hope I can give you just a flavor of how complex these proteins are. So here is the gene that basically encode this protein. So there are 3 different genes. There's a light chain, there's 3 heavy chain involved in one protein, or there is 2 variation in the blood that we can find. So one is called inter-alpha inhibitor, the other one is pre-alpha inhibitor. Initially, people think that is the precursor, but actually it's not, because the heavy chain is a different. So there are 3 different places of the chromosome that regulate this, or express this protein, located the gene that express this protein. So this is the 2 protein, almost like 98% that circulate in our body. So, and we call this a family. So it's inter-alpha inhibitor proteins, plural, because it's always together somehow, the 250 kilodalton and 125 kilodalton, although it's 2 different proteins. So the only things that the same is the light chain. The light chain is present in the larger molecule and in the smaller molecule. So when there is an inflammation, there is a cleavage here. The light chain is released, and when it's released, it goes to the kidney, filtered out, and you can find it in the urine. So this the light chain is called also urinary trypsin inhibitor, because it's still active as an inhibitor in the urine. So in Japan or in China and Korea right now, people produce this from, basically collect the urine from mostly pregnant women which excreted more of this protein, and they isolate it as a drug. So, and basically also called biquinine. The reason is because there is 2 different domain that they call it Kunitz domain, that basically active in the active site of this inhibitory activity. So that's why they call it bicunin, because of Kunitz domain 1 and Kunitz domain 2. So so this is the one that excreted in urine. There is 145 amino acid polypeptide that basically this is the one that give the activity of inhibitory activity of inter-alpha inhibitor. So not the heavy chain, just this light chain. And I mentioned before, it basically block a lot of serine protease, if not all. So basically like a trypsin, elastase, plasmin, chymotrypsin, catepsin, and granzyme K, and furin. So furin is interesting. Reason is because of the COVID-19. So it turned out furin is the proteases that in our body. So however, so this furin involved in many processes in our body to cleave hormone and and other stuff. So if it's basically changed or pathologically active, so it can cause cancer, diabetes, inflammation, and neurological diseases. So this furin is over, it's available in the body, in every organs, and so on. So, but this furin enzyme in our body is used by virus or by bacteria actually. So because they don't have this furin, this enzyme, so they use this enzyme to process. So how important is it's actually pretty important. So as you can see, this is the SARS-CoV-2, the current pandemic cause of the disease today. So as you know, there is a spike protein, coronavirus with the spike. So the spike actually has this on the S1 and S2. So the S1 and S2 is part of the spike protein that has to bind to the receptor in the cells. So the lung cells and so on. The lung cells mostly has this ACE2 as a receptor for this. So it's an angiotensin enzyme that basically bind as a receptor of this coronavirus. However, furin has to remove the S1 before the S2 can bind to the receptor. So with the S1, it will not basically get tight interaction, and the virus cannot get into the cells. So only with the help of furin, when the furin eliminate or remove the S1, then the S2 can bind, and then the virus can get into the cells. So that actually, people trying right now to find an inhibitor of furin. Maybe it can block the entry, because the only way the virus can replicate is if they get to the cells and use the cell machinery to replicate. So in this case, not only coronavirus, a lot of viruses use this. Even herpes virus, retrovirus, and recently even flu virus use this furin, and also bacteria actually. So the reason I'm interested in this is because a couple years ago we worked with anthrax. So anthrax toxin, the same way, use furin as need help from furin. So yes.
Yeah. Can you back to the slide before this? Yeah. Okay. The furin about
furin. Yeah.
Yeah. Okay. This is I think it should it is very interesting, but I need to or maybe we need to have a little bit discussion, a short discussion on this idea. So furin is a protease, yeah? Serine protease. Is it called furin? Yeah.
Yeah, furin is an
Yes.
enzyme. You know,
Yes.
it's
And
it cleave. You know, that's why it's
Yeah.
depicted as a scissor here. It cleave other protein.
yeah.
So in this
And
case, you know, like cleave this S1 and S2.
yes. This inhibitor, furin inhibitor, inter-alpha protein inhibitor is belong to the furin
Well,
inhibitor.
that's what so that's the interesting question. So I got interested in this, and when I saw, you know, that the coronavirus need this, so we doing some experiment. But so before so this is the one study that we published this. This is in 2005. So we title it Inter-alpha inhibitor proteins are endogenous furin inhibitors. And it was done in anthrax toxin. So it's very similar story. So anthrax toxin here is depicted as PA83. So they call it, you know, protective antigen 83, and there's also another lethal factor. So this has to come together and basically, you know, before it enters the cell to kill the cell. So, but the furin has to remove some part of this 83 to become, you know, 63 and 20. So without that cleavage, the furin will, without the furin cleavage, will not, the anthrax toxin cannot get into the cells. So it's very interesting that you know, the bacteria or virus use our enzyme to help them to gain, to, you know, to kill us, actually using the weapon that is in our body. So so basically what I'm trying to say is, you know, inter-alpha inhibitor actually inhibit furin as well, that may be involved in the coronavirus processing. So we have some preliminary data that I don't want to share it here right now, but it seems like it's similar, like the anthrax toxin. It blocks. So we found here in this paper, you can find it, you know, that we basically increase the survival of the animal that we injected with anthrax toxin, the inter-alpha inhibitor. So yeah, so that's one of the you know, inhibitory activity of the protein, but much more than that. So because the you know, the protein is not only as a protease inhibitor, but other things. So that's why I wanted to show you the complexity of these proteins. So anyway, so that's the light chain, which is, you know, the active side of the serine protease inhibitor. What happened with the other part of the molecule? We call it heavy chain. If you remember, there is 3 different heavy chain that attach to this protein. So and the unique part of this is, you know, there are 3 that is major circulated in the body, and the heavy chain and the light chain linked by sugar molecules. It's very, very rare that is, you know, 2 proteins are linked by glycosaminoglycan or sugar chain. So one of the very rare protein is this inter-alpha inhibitor. But in any case, so there are 6 known of this heavy chain, and all has this characteristic of, they have a VIT, fault protein inter-alpha trypsin domain. So this is characteristic of this family of protein. And there's another one that every single member of this heavy chain has. It's called von Willebrand factor A, which is very, you know, it gives this characteristic bind to many different substrate, like hyaluron, complement, collagen, integrin, and recently we found that it binds histone, bind HMGB1, which is a damn or dangerous protein. It's because we believe it's because of this domain here, the von Willebrand factor A domain that bind very broadly to many different substrates. So, and that's what gives this protein very unique characteristic, I guess. Any question, or should I continue? Please, if there is a question,
Yeah.
I don't mind at all to interrupt the. If not, then I will continue. So it started actually, we have a monoclonal antibodies, and then we develop a immunoassay. So this is like a tool to use it as a way to quantitatively measure the level in blood, in urine, and so on. So we have this assay initially established. So you can see there's a standard curve. So we can measure an unknown sample, and then we can determine what's the concentration of it. So the very study that we did is we look at the level of this protein in sepsis patient. This is patient in the ICU that died or survived from a sepsis, and compared to the healthy control. So we have, this is very, a long time ago, we published in 2003. So, but interestingly, we saw that is like a gradient. So when patient died, basically the level of this protein is very low compared to the normal. So it's lower than the survived one. So, and then we look, this is a bacterial sepsis. So we look at dengue viral infection. So what happens if you have a dengue, so viral diseases that also, very similar, can create a dengue shock syndrome, which very symptom is similar to the bacterial counterpart, right? So, and this is actually in children, and indirectly we get the samples from a collaborator in Holland and in Netherlands, and they get the samples actually from Indonesia. So indirectly we study Indonesian children with dengue here. So we published in 2010. We saw the same pattern actually. It's normal, and if you have a dengue fever, it's lower, but the lowest is when you have a dengue shock syndrome. So with this, so we try to use as a biomarker. So maybe we can use it to detect or to use as a host response biomarker to give a doctor idea whether somebody who has the low level is need much more attention and so on. So we are developing right now the rapid test. So this is a ELISA. It takes like 6 hours in the lab. So we are right now developing a rapid test that can take only 15 minutes without any equipment. So it's similar to the IgG or IgM COVID-19 serological test. So use a lateral flow. So we will shortly have this test. So basically you don't need to have a skill to do it in ELISA format, but we can do it so in the bedside test. So testing in the bedside without any equipment. So, and we look at, so that was an adult or children. So what happened in the newborn? Actually, we found a similar pattern. So we have here's a blood negative culture babies that negative of bacterial. So has much higher level, but the positive blood cultures is much lower. So, and then we look at not only sepsis, how about necrotizing enterocolitis? This is a very, very, very deadly disease in newborn, that babies that born prematurely, they usually get the necrotizing enterocolitis, and it's very rapid. Babies died within, hours if after they diagnose. So, and we see also, the control is higher than the babies that proven with this disease. So, and here just to show that we can actually, in the babies, we can use it as a diagnostic tools, because we can detect we can identify the babies that has this necrotizing enterocolitis or neonatal sepsis. 98% accuracy, and compared to the C-reactive protein called CRP, it's only 65% to sensitivity to detect this. So maybe there is a potential to be used as a diagnostic test as well, but only in the babies, not in the adult. So after completing that study in plasma, this is in patient plasma, so we thought like, wow, so if the level go down up to 90% of the normal level, so the next question is, what happens if we basically normalize it? So give this back to the patient. So we cannot do that in patient, but we can do it obviously in animal study. So that was the past 10 years we have been doing many different model. So one is a sepsis model of Cecal, ligation, and puncture, and then anthrax and lung injury and so on. And lately we did actually stroke as a part of inflammatory disease as well. So I will maybe show some example of this. So here's just to give you an idea how the model of sepsis are done. So this is actually the gold standard in the research community. They call it Cecal, ligation, and puncture. So basically, it's just a mouse or rat. So, and then you surgically open and remove, take out the cecum, and then you ligate it, the cecum, and then you puncture with a needle. So, and it's interestingly, it depends on how big the needle is, and the mortality is correlated with that. So once you puncture, you squeeze the content, and then you basically close, and you can imagine that it will create a peritonitis in the cavity, and that's where we create this, or induce the sepsis. So within 1 or 2 hours, you will find bacteria in the blood when you do this Cecal, ligation and puncture. So, and if you don't do anything, so they all died within 2 days. But at day 2, you can open and then remove the ligated cecum, and because now it become already necrotic, so when you remove it, so 30% survive of the animal up to 10 days. So that's our standard study. So we induced sepsis, we took out the necrotic cecum, and then we continue to watch the animal to survive 30%. Most 70% will die. So that's the model. So this is the initial study that we did. So we purified this protein, knowing that this protein is really, a lot in the blood. So we didn't do it in the urine because the urine is only the light chain. We wanted to have the whole molecule, which is, with the heavy chain, which is only in the blood. So we isolate this from the blood, and then we give the animals, we challenge the animal, and then we inject the purified inter-alpha inhibitor, 30 milligram per kilogram body weight, and then watch within 10 days, to see, to calculate the survival rate here. As you can see, when we give it 1 hour after that sepsis. So 1 hour, because at 1 hour, per definition, you can find that bacteria in the blood. So, and we can increase the survival from 30% to 90%. So when we delayed, not giving immediately 1 hour, but we delayed to 10 hours. So 10 hours in this animal, it's maybe equivalent to a severe sepsis in human, whereas 1 or 2 organ dysfunction already happened. So severe sepsis, so not to shock yet, but it's already like 1 or 2 organ dysfunction happen. So even that, so we give the inter-alpha inhibitor late, so we still can rescue almost the same rate as early. So we thought this is very interesting that maybe inter-alpha inhibitor is protective. So why? That's the question. How this molecule work? So that's actually the focus of our study in the last 5 years to understand how come this protein has really this protective effect. So and that's why we basically discovered one by one the effect of this. Maybe not only the light chain, is the heavy chain is even more important than the light chains that actually contribute to this protective effect. So here's another study that we use. We compare. Well, we study the endothelial inflammation. So this is a sepsis patient that we collected in the clinic, and then we look at the inter-alpha inhibitor level, and then we look at the FCAM and ICAM. This is 2 molecules as a marker of endothelial inflammation. So it seems like there is a inverse correlation when the inter-alpha inhibitor is low, this inflammatory marker is high. But if its inter-alpha inhibitor is high, it's very low. So there is a somehow indication that inter-alpha inhibitor maybe play a role in protecting the endothelial, which is important in sepsis and so on. So another study is, this is a mouse study that we injected inter-alpha inhibitor, and then we labeled it, and so that we can see the color of inter-alpha inhibitor is red, and hyaluronan is green. Hyaluronan is another substrate that maybe bind to the heavy chain of inter-alpha inhibitor, and it seems like they are together. So this orange color that show that is basically when we merge that. So it's basically bind together. So inter-alpha inhibitor basically co-localize with hyaluronan and maybe protect the endothelial cells. So in this study we did animal models and so on that showed the effect of that. So endothelial cells is important in systemic inflammation. So, and then to our surprise, so we did something unexpected in the brain. Actually, we found that when we study, there is a reactivity, using the antibodies, we found that the inter-alpha inhibitor is detected in the brain parts, so in the cortex, hippocampus, and choroid plexus, and so on. So even and the cells in the neuron, in the microglia, astrocyte. So this is really a surprise to us because this protein is mainly made in the liver. So we do not know why this protein is expressed in the brain unless there is a role in there. So we believe maybe it's as an immunomodulator in the brain. So that's the finding that this is maybe 5 years ago we identified, but then we study stroke. So if we know this protein is in the brain, maybe some effect on that. So basically we have a model that it's called middle cerebral artery occlusion. So you basically open the artery and then basically you ligate the middle cerebral artery so that the blood flow disrupted. So and if you do that, so this is the young mice, and we did also in the aging mice. So this is very different, when young animals where everything is still good, but in aging mice, 18 to 20 months old is about equivalent to 70 to 80 years old person. So everything is like the artery already brittle and so on. So we need to do this because it's different physiological background. Here, same like in people. So with the age, you have hypertension and all the problem with the everything that basically affecting because of aging. So and young mice usually don't have a problem, but so when you do the ligation, so you will get this. So basically, a white area which is it's an infarct. So that's the infarct site or infarct area. So when we injected inter-alpha inhibitor, we see much smaller infarct size. So this is the area that in the cortex, in striatum, or in the total. So basically from 50% down to 20%. And we did also a lot of behavior study on this because it's the mouse. So you can study how the mouse behave after you challenge with this stroke. Basically, you can see how they memory and so on. So it's all basically with the inter-alpha inhibitors improve. So we are preparing the manuscript we will publish soon. So and the aging mice. So some drug that work only in the young mice, but when they try with the aging mice, didn't work. So as I mentioned, because there is a much more complex underlying differences between the young and aging mice here. So here again, so when we inject, this is the control animal injected with only the buffer compared to the inter-alpha inhibitor, so much smaller. So we think that this may be inter-alpha inhibitor protective if in the brain as well. So because at the end of everything is inflammation, so in the inflammatory process is maybe stopped by the inter-alpha inhibitor. So that can be useful in this disease. So and then we look at the study in stroke patient. So we just measure the inter-alpha inhibitor level. So this is the stroke patient. So there is it's a statistically significant drop, and this is 24 hours after stroke we measured this. So, and also we have an assay that measuring inter-alpha inhibitor with histone. I will get back to this, why we measure the histone. So, and there is totally increased level of the histone and inter-alpha inhibitor complex. So why we see this? So we know that, so this is a cell, has a nuclei, whereas all the chromosome or the DNA is basically located, and actually, so in between the DNA, there is a histone proteins, basically just like what do you call that, a benang. So it's like a spool and connected each other, but so the histone is maybe protecting the DNA. So, and histone, there are 5 different types. So one is H1, H2, H2B, H3, H4. And this is, the whole thing is called nucleosome. So the body never see histone. The histone is only in the nuclei of all cells. So we never see histone, but when the cell died, this histone is released. And that's why we call this histone danger signals or DAM, because it is actually become problematic. It can cause inflammation and thrombosis actually. So, and here's the study that we did with when we immobilize histone on a plate. So, and then we add inter-alpha inhibitor. So, and we know that inter-alpha inhibitor bind to almost all, with preference of the heavy, the H1, the histone 1, but almost all bind. So we know that the histone actually bind. That's why we measure in that study, previous slide that we found in the stroke patient. So basically, as with, cell died, so stroke means the oxygen cannot go to the area where it supposed to go, and the cells without oxygen die. So when the cell died, it spill over the histone, and the histone basically looks like it catch or complex with inter-alpha inhibitor and neutralize it. So and become less problem for the body. So, and we published this actually is in 2015. So and here's again just to give you a cartoon here. So immune cells or any cells can get apoptosis and so on. So the cell death is different cells, different processes. They call it mitosis, apoptosis, pyroptosis, and necrosis. So it could be immune cell, could be parenchymal cells, liver cells, or brain cells. So when it died, it released the histone, and then basically cause the thrombosis or platelet aggregation, and that's basically what in sepsis happen in the small capillary. When you have a thrombosis, then become DIC, disseminated intracoagulopathy, and that's part of the shock syndrome. So, and here's, so when you have, so we try, this is in the animal, so we inject histone alone, and then measure the platelet consumption. So when we inject the histone, the consumption is 80%, but when we give inter-alpha inhibitor, basically the consumption is less, half. So and as a control is this is hyaluronic acid, which is already known bind histone as well. So this is the positive control. So inter-alpha inhibitor behave like the hyaluronic acid, so to protect the body. So and when we measure the bleeding time, this is the tail bleeding time of the animal. So the control is, it's normally a few seconds or minutes, sorry, 2 minutes. So when you inject histone, the bleeding time is up to 10 minutes. So when we basically treat with inter-alpha inhibitor, the bleeding time back to normal, same as in the histone. So, and this is the lung tissue injected with histone. You can see the control is clear. So there is a microthrombi, so it's fibrin rich thrombus that detected in the lung. So, but it's not in the inter-alpha inhibitor control. I mean, treated animals and so on. So again, this is published already. So seems like, you know, the histone is part of this the important factor that cause problem in systemic inflammation. So actually, you know, when you think about this histone, so in the cells is okay in nucleus, but as soon as become extracellular, that's when it become problem, and it has been implicated in stroke, in lung injury, in, toxic liver injury, acute kidney injury, pancreatitis, peritonitis, appendicitis, and so on. So you can see, you know, anything that has a cell death, it will create histone outside the cells, and so it affects many different processes or diseases. So that's one called histone, but there are many others. So, and recently this is the second one that we found, a molecule called high mobility group box 1, very odd names, but it's called that. It's a protein, and it's a nuclear protein. It's very similar to histone. So only in the nuclear, but it will spill when cells died and so on. So this is just to show a similar study that we prove that actually this molecule, both molecule, inter-alpha inhibitor and this danger signal protein actually interact. And this is, we have a solid binding assay similar like ELISA, and we did also with the surface plasmon resonance technique that you don't need to label. In this case, we labeled the protein. This one doesn't need to use label because it measured the like a laser angle. So basically, you can conclude from both assay that it is specifically these two proteins interact. So when you see this is this is in vitro. So this is cells in the brain, actually in the cortex of rat brain. So the red one is HMGB1. We fluorescence labeled antibody against this protein HMGB1 that is red. So, and this is DAPI color. So any cells will show blue. And the inter-alpha inhibitor, we label it with the green fluorescence. And then we basically merge the two, and you can see this purple color. So the combination of two, that means that they are located together. So this is in vivo. So this is in vitro, this is in vivo. So we haven't shown that it's really neutralized the effect of HMGB1, just like histone in the previous slide. So we are looking into a way that we can show that really, you know, the binding or the interaction of the two protein outcome is actually to reduce the inflammation or neutralize the HMGB1 effect. So HMGB1 actually bind to a toll receptor. So this is again danger signal. It dangerous signal because it bind to a surface receptor, and when it bind, and then the cells produce a lot of cytokine. So and hopefully inter-alpha inhibitor block that. So. And this is the last part that I wanted to mention. It's very exciting because it's unexpected as well. So this is something to do with neutrophils. Neutrophils is a part of the white blood cells. So it's considered immune cells. So when we have neutrophils, as soon as you isolate neutrophils, usually they are in this form, very irregular, because that is the activated state. When the neutrophil is activated, they form irregular. So as you can see here is the scanning electron microscope. So this is in the buffer solution. So it's really irregular. So when we add human serum albumin, doesn't change anything. Bovine serum albumin doesn't change anything. And there's a protein that's known to change to basically cause the neutrophil to resting state called histidine-rich glycoprotein. So this is a control for us. So when we add inter-alpha inhibitor in different amount, as you can see, it becomes rounder and rounder. So when it's around, it means it's not active. So basically, it's in resting state. So here's the fluorescence study. So you can see again with different amount of inter-alpha inhibitor. But when we add the light chain, which is called bicunin, it doesn't seem like doing anything. So we believe that is the effect. This is maybe the heavy chain. So the light chain is good for inhibiting the proteases, but the heavy chain works something much more than just the activity. So again, inter-alpha inhibitor maintain neutrophil in the resting state. So here's just you can also measure the form factor and so on. So basically, if the diameter, 1 is perfect round, so 1.4 is oval, irregular, so it become more and more near 1. So
yeah, Prof. Lim.
Yes.
So the time is up.
Yes. Okay. This is almost done. This is almost done. So this is the last.
Yeah. Okay.
Okay. So let me. This is just to show the consequences of that. Why, if it's round, it can go very easily to capillaries. So we measure. Here's the control. Basically, if you have bicunin, is the cell stuck on top, you cannot go past this. So there's a movie published here with this in this journal. So again, the production of reactive oxygen species also reduced by the neutrophils that show that reduction of inter-alpha reduction of the inflammation mediator. So seems like a lot of things that happen. So here's the, summarize this. So the light chains inhibit the serine proteases, but the heavy chain actually did much more. So upregulating the anti-inflammatory mediators, down-regulating the pro-inflammatory mediators, and then block complement and block danger signals that, the two proteins that I saw, and then protecting the endothelial cells, and lastly, reducing the neutrophil activation, and that reduce also the cell addition and mediator production of ROS. So all together, actually, that maybe give a unique effect of inter-alpha inhibitor. So in summary, so I just wanted to take the last step here. It took more than 15 years extensive study of a protein to get ready now that we know more about this. So we have a partner that will manufacture this of protein to a clinical grade that we can use it to test in clinical trial. And so with that, so I wanted to show the thing is still not done. We are just in the middle of this. So we are finishing the basic and preclinical research. We are now entering the product development. So we have to show the manufacturing process is stable, and consistent and so on. And then we can enter phase 1, phase 2, and phase 3, and the final, the finish line is the FDA approval. So it will take still a couple more years. So, but I just want to show this as a, reminder that it takes a long time to develop drugs. Not only, take time, but also money, costs a lot to do the research. So it was estimated, up to $800 million to develop a drug from scratch. So it is really, many years and many collaboration. So this is what I wanted to finish. So a lot of it's a collaborative effort. So it needs to be, a joint, work with immunologists, pathologists, neonatologists, stroke specialists, and so on. So I just wanted to thank this individual and NIH, which actually support all the study with the grants. So with that, I close now. So sorry if too long.
It's okay. Prof. Lim, we need to be efficient with our time. So there are several questions on the chat from the chat.
Yeah.
So we want to share. We want to show you the question. Yeah.
Okay.
Yeah. So, can you read?
Okay.
Okay. Yeah. These are the questions. You may
the protein
respond. Yeah.
Okay. So I didn't study furin a lot. So what I study is only in anthrax. So in avian flu, it's true. It basically cleave the hemagglutinin and so on. But in cancer, so when something is not right, so the furin not work properly, so that's when it causes cancer. So I cannot answer specifically in this thing because I'm not working on cancer at all now. I used to. I started with the cancer study, but so I cannot answer this question. So it's possible that the protein has a crosstalk by suppressing inhibitor of apoptosis. It's possible, but so furin is only an enzyme, very common enzyme in our body, in every organs and so on. So it's possible that protein has a crosstalk by suppressing inhibitor of apoptosis. Talking about inter-alpha inhibitor or the furin here? I'm not sure the question. Does it activate another specific protein? Well, so basically if it inter-alpha inhibitor, yes, inter-alpha inhibitor works so many things. So again, so the apoptosis may be blocked by the way of, so just like you know, like the apoptosis, the necrosis and so on that I showed before. So it's indirectly. I think it's indirect effect, not directly to the apoptosis itself, but indirect to other proteins. So something that causes apoptosis but is blocked by the inter-alpha inhibitor. So second question, is furin used by numerous infectious diseases to enhance infection and replication? Yes, that's the idea. So the bacteria and virus do not have this mechanism to cleave it. So actually, they need our help to actually become, you know, a culprit for our body. So it's very ironic that we help them. Identifying counting protein in the blood is a long process. Do you find it easier now with advanced technology? So identifying and counting protein. So actually, to identify a protein, there's a method called mass spec. So there's a lot of people already done work and map, you know. So what if you do not know the protein? So you can use the mass spec and then compare to the database. Most likely you will find you know, that your proteins may be related to certain things in the database. So that's how we used to do this, identifying a unknown protein. So that's the advanced technology. That's correct. So there's a mass spec. There's very, very advanced things now, you know, and then proteomics, for example, studying all the relationship with protein, different proteins, you know, global proteins. So basically it's use mass spec as well. So to identify it and then see the interaction between one another. So we call it proteomic study. So that's the advanced technology. Immune system can use inter-alpha inhibitor as endogenous inhibitor. It is possible to get immunity through diet that can produce furin rather than urine. No. So the furin is the one that used by the virus. So so whether, yeah, I don't know whether if you eat, you know, certain things can produce furin or not. I'm not sure, but again, furin is a enzyme that is important for your body. So it's nothing to do with urine though. Would histone protein be released extracellularly in an apoptotic process? Yes, which highly organized by the formation of cell membrane bubbles to prevent inflammation. Yes, actually, so that's the idea behind it. So histone release, so the cell undergo apoptosis, so cell death, and then release the content, which is, you know, the histone and the DNA. So the DNA and histone supposed not to be seen by immune cells in circulation. So and that's why, you know, a problem. So we should prevent the histone. So that's one of the things, is maybe inter-alpha inhibitor can neutralize that. So I'm sure other protein can do that too. So some people make antibody against histone, so that neutralize it. So so the nice thing about inter-alpha inhibitor is it is a natural. It's already in your body. So I don't know if that answered the question or not, but hope I answer it. But if not, yeah, please ask again.
Okay. Maybe there are direct questions from the participants. So participant, maybe 1 or 2, 3 participants who want to ask directly to Professor Lim. You can raise your question. You can unmute your mic, and then you can ask directly.
Okay, good morning, Professor Lim and
Yeah.
Professor Chris.
Yeah.
My name is Richard Steju, and I am actually in great interest on your presentation about how IAIP and inflammation interact. And then I have some concerns, Professor Lim. Sorry. Yeah, since furin is also essential for cellular function, will the administration or IAIP later on will disrupt, the normal process of the molecular, cellular, or organs?
Yeah.
Let's say we choose the IAIP as a treatment for inflammation.
Good question. So I think it's just temporarily because the furin is in your body, the inter-alpha inhibitor in your body, but the problem is when you have an inflammation, sepsis or anthrax or COVID-19, the inter-alpha inhibitor goes down. So and when it goes down, so there's nothing that the balance somehow changed. So now the COVID-19 can get into the cell, the anthrax can get in the cell even more. So I think it's about just temporarily. So that's the reason why I think is great about inter-alpha inhibitors. It's just like we are just trying to replenish or to correct or to add back to what you lost before. So I hope it answered your question.
Yes, that's clear. So that means that we need a certain dosage to offset the lack of the amount of protein during a pathological state then.
Yeah, so that's the study that we will need to do is the dosage study. That's the clinical trial. So
Right.
The first thing you have to show safety. So that's the phase 1. And then once it's safe, then you do basically this dosage study. How much is can you give to that is tolerated by the patient, but not toxic, but also have an effect that you want it to get. So that's basically the study for the human testing. But in the animal, we have done, of course, you titrate. So we found that 30 milligram per kilogram is basically the optimal dose. But so animal, mouse and rat is not human, so it's totally different. So has a so there is an equivalent dose, but so we have to test that. So somehow we know already how much to start with, but so we will do it the titration there in the clinical setting.
Right. Okay. Thank you, Prof.
No problem. Yeah, very
Thank you.
very good question.
Thank you for the question. Yeah, thank you for the question, Mr. Sutejo. There is a question from Dr. Marty Santoso.
Yes.
Is Dr. Marty Santoso still active in this webinar? So you can directly raise your question.
Okay, yeah, I can read. I think
I can read the question.
Yes.
In my clinical practice, we use procalcitonin and
PCT.
CRP as a predictor the infection becomes sepsis. How is your opinion there?
So procalcitonin is only increased in bacterial sepsis, not in the viral. So that's the difference between the inter-alpha inhibitor with procalcitonin. Procalcitonin seems only bacterial sepsis. So CRP is more general. It's like it's even so very non-specific. So I hope that we compare the CRP with inter-alpha inhibitor in one of the study in neonatal sepsis, and we found that CRP is very, like 65% detected power, but inter-alpha inhibitor is around 90+%. So procalcitonin is nice too. So it's really good. The only thing is maybe not as a broad inflammatory marker. It only affects bacterial sepsis.
Yeah, but this is but an alpha inhibitor protein is not specific for viral infection.
No, that's right. So the inter-alpha inhibitor, when it's go down, it just shows that your immune system or your innate immunity react. So basically it's showing there is a host response. So same thing like fever, right? So if fever is caused by TNF alpha, it goes up. So the TNF alpha is basically caused by secreted by immune cells and so on that give you the fever. So in the same way, inter-alpha inhibitor also gives some thing, so not all infection give inter-alpha inhibitor. So local, so for example, we have a meningitis. We don't see any increase or decrease in inter-alpha inhibitor because it is local. So we are talking about only if it's become systemic. So when it's become systemic, then inter-alpha inhibitor is affected. So fever is nice too because it shows that you have some reaction of your immune, but you don't know where it start and so on. So many symptoms, right? So whereas inter-alpha inhibitor, maybe you can say your host your immune system is challenged here, and when so the what I try to do is there any like level that you can show the lower the level can show that you are more dangerous than so basically can be used as a indicator how bad your situation is, something like that. So we are currently studying the COVID-19 patient in the ICU or in the emergency room. So the mild, and the severe one, and then see the difference of the level. Yeah.
Another question. What opinion in your study? This is important if we can detect earlier.
Yeah.
Maybe 2 or 3 days. This is a report for the clinical.
Yeah. So we did study in the neonatal sepsis. So what we found is we published this. So the blood culture started, later, actually 2 days before the blood culture positive. Inter-alpha inhibitor actually already low. So we conclude that maybe it's earlier than the blood culture itself. So it's because it's the body is challenged, so the immune system is maybe more sensitive than the detection of the blood culture, in the blood itself, because it's a lot of positive and negative result as well, right? A false positive or false negative with the blood culture. So we think that inter-alpha inhibitor maybe is a good early indicator. So as soon as your immune system is challenged, it maybe should go down. So the only problem is everyone is different. So that's why I think the best way is to measure. So the ideal situation is in the ER, in the emergency room. So patient come, the doctor don't know, so take the status of the level. So if it say 300 and an hour later it's 400 or stable, so it means maybe it's not that dangerous. But if it's from 300 and then become 100 within a short period, then you the red flag. You should really treat the patient very aggressively. So that's maybe the value of this assay. So mainly not to diagnose disease, but to give you a prognostic indication.
Yeah, like a predictor. This is good.
Predictor, yes.
Yeah, if the study can be applicable in the clinical practice, it is very good, because sometimes we also confuse this become sepsis or become worse this infection. Thank you, Prof. Lim. This nice lecture. Good lecture.
Thank you.
Welcome.
Thank you for the question. It's a good question.
Yeah. Thank you very much.
Okay. Thank you, Dr. Marty, for your question. Is there any participant who want to ask to Professor Lim So I will try to offer another participant. If there is no other question,
sorry,
okay.
I would like to ask. Sorry,
Okay.
it's me again.
Please, please.
To continue with the previous question, you are mentioning that it is not for diagnostic from Dr. Mardi, but I saw just now the AUC curve is even covered at 98%, Professor Lim.
No, that's so I distinguish.
That?
Yeah, I distinguish between the adult and the children. In this case, in infants. So in infants, it's very clear. So that study was done in infants. So we believe maybe it can be useful to diagnose disease in the infant. But on the other hand, the level actually very low in sepsis and also very low in necrotizing enterocolitis. So per definition, if you wanted to use it as a diagnostic tools, right, you cannot do that because both are low, so it's not specific which one. But on the other hand, those two are very, very related. So the patient that has necrotizing enterocolitis, it will get sepsis. You know, so basically at the end, maybe it's not big difference. But if you strictly, use the definition of diagnosis. So diagnosis is to diagnose a disease, specific disease. So this protein or this marker may not be useful because it's just showing you are in very dangerous situation if you are in.
But we cannot specify what because it's too broad.
Yes, yeah, and
okay.
So the problem is, especially I work along with the neonatologists, so they don't have tools to know a baby has even harder. In babies, it's really harder to have the symptom, right? So like fever or whatever is so confusing. So even more important to have that indicator, something that is really, really, dangerous or not. So especially, children with, so a couple of years ago, there's a case which is printed in New York Times because it become very, 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 headlines because a boy, 10 years old, died of sepsis. Actually went to a very, very prestige university hospital in New York City. So, and it died because the symptom is basically just like fever. It's, throwing up. So they give a fluid, they give a Tylenol and send home. And the next day it really, came back with basically need to intubation. This is 10 years old, very healthy boys. So and so because the problem is there's no indicator that all symptoms are the same. Blood pressure low or fever and can mean a lot of things, right? So that's maybe the value of intravenous.
I see. Okay.
And pinpoint. Oh no, this is actually much more integral part of your host response, inflammatory response, and if it's really, really low, So
it's only an indication of very general pathological condition, but to pinpoint or to a certain disease, then it's not possible then.
No, yeah. So if it's a pinpoint a disease, like certain disease, cannot.
Yes.
So because it's not the protein is not produced by a bacteria or something. So that's the only way, right?
Right.
Do if it's like COVID-19, if the spike protein, so you can say yes, the spike protein is there, then you know it is a coronavirus. So that that's not this is not diagnosed to diagnose disease. Yeah.
Right. Okay. Thank you, Professor.
Okay.
No problem.
Yeah. Thank you very much. So, Professor Lim, it is possible to detect inter-alpha protein in saliva?
Yes, we did actually. So you can level. So basically, so the question is we haven't correlate the level of inter-alpha inhibitor in the saliva and the blood, but you know, I study from other literature, you know, mostly say that saliva is extension of it's extension of the blood. So, but I don't know, you know, whether it changed, because when you drink, you more dilute and so on. So it's difficult. That's the reason why we do not use the urine as well. So because, you know, urine change, the more you drink, the, you know, the more dilute and so on. But blood is very, very consistent.
Is there any kit available at the moment for detection of inter-alpha protein inhibitor?
No, we are working on it. So, so we will work. So maybe soon, you know. So we have a partner. So Takeda is the one that we're interested in the therapeutic protein itself. So they are blood product company. So one of the part of the Takeda. So we are working with them so to do this, but also developing the assay as well. Yeah.
And that means that the kit is ready ready for clinical trial.
That's what. So I'm working on it. We have a grant to transform the ELISA format to 6 hours to 15 minutes. So the technology is very simple, actually. So it's exactly the same as the COVID-19 serological assay. It's basically a lateral flow. You don't need a pump. You don't need, you know, it's basically one drop plasma or serum, and then let it run itself. And then there is a line which is, you know, similar like a pregnancy test, but the pregnancy test is yes or no. So we are trying to do very, very, you know, quantitative. So accurately can say, you know, if this is changed to this color, this is the level, and so on. So there will be a small reader that read the intensity of the bands or the line. So and correlate the more the intense the color, the higher the level, something like that.
Are you planning to have clinical trial in Indonesia?
Well, so let's see how it goes. But you know, so the idea is, you know, the FDA usually only recognize certain, you know, level of like the quality control things. So if you do it in Indonesia, you submit it to the FDA, they will say, well, you have to repeat it in here. So
Okay. Before we close our seminar, we need to make photos for all participants.
Oh.
So please unmute your computer so that our staff may make photo for us. So then I would like to thank Professor Lim on behalf of all participants and also on behalf of UKRIDA. We hope that in the future we may have another lecture with another topics, especially for the student, for the basic understanding of information and so on. So that is, I mean, medical student. So,
thank you.
Tolong diundang lagi, Pak. Profesornya tolong, Pak. Kapan diundang lagi, Pak?
Ya, kapan-kapan nanti saya undang lagi ya.
Kalau bisa dengan kolaborasi berbagai FK, nanti saya dari Trisakti bisa koordinate, Pak.
Ya, bisa juga ya. Bapak tahu alamat email saya ya, nanti kontak.
Saya belum tahu, Pak, tapi saya ada HP saya.
Ada.
Kan?
Ya.
Yang Gunawan Cahyadi, Pak.
Ya,
saya dulu pernah ngajar
Gunawan Cahyadi di mana? Di UKRIDA juga. Ngajar di mana sekarang?
Saya pernah ngajar di UKRIDA, Pak, tapi bukan di FK, di elektronya, Pak.
Elektro
Dengan
ya.
Pak Samuel dan Pak Albert.
Pak Albert.
Tapi saya megang biomedic
Ya,
engineering sekarang, Pak.
Iya,
Makanya
iya,
saya
iya.
join this session, Pak.
Iya.
Pak Gunawan di mana di Trisakti?
Iya, saya di Trisakti, tapi 2 tahun lagi saya pensiun karena sekarang saya umur 63. Jadi sebelum saya meninggalkan kampus, saya mau kasih sesuatu lah buat kampus saya, Pak.
Ya.
Very good.
Zamannya sekarang kan kolaborasi, Pak. Sudah nggak
Betul.
boleh kita bergerak
Ya.
sendiri-sendiri, Pak.
Ya,
Pak ya. Bapak
Ya,
mungkin
jadi
setuju sama saya.
Mestinya bisa kolaborasi ya. UKRIDA,
Iya, betul.
Trisakti,
Dan kita
Taruman
harus
Negara
ingat,
yang
Pak,
dekat-dekat.
kita hidup di masa lintas ilmu sekarang, Pak.
Ya,
Sementara
ya,
kita dosen cuma bisa kasihnya monodisiplin, Pak.
Ya,
Itu maaf ya, Pak, yang saya harus bilang terus terang.
Nanti
Pengalaman saya ngajar 38 tahun, Pak.
ya. Ini untuk virus juga ada teknologi nano biosensor ya.
Iya, betul. Saya
itu
megang
menarik
kuliahnya
juga.
sekarang nanoteknologi dan
nanoteknologi.
opto, Pak, untuk
Opto,
di semester akhir, Pak.
Ya.
Iya.
Oke lah.
Mungkin
nanti kita bisa kontak, bisa ketemu lah.
Baik.
Ya,
Bapak bisa korek saya dari staf elektro Bapak lah.
Ya. Nanti saya di Tanjung Duren
Pak.
ini. Ya.
Terima kasih.
Terima kasih.
Ya.
My appreciation to you, Professor.
Ya, untuk kita ketahui, Prof itu lahir di Cirebon ya.
Betul, saya dari Cirebon aslinya.
Dari Cirebon.
Prof ini temannya Bapak Lim Yosin ya, atau So Siteng ya, betul?
Yosin itu kakak saya.
Oh my goodness, dulu Pak Yosin sama-sama saya di Untar, dia di UPH.
Betul.
Iya, terus dia migrate ya kalau nggak salah ya, Pak.
Dia di Singapura. Ya, di Singapura.
Iya betul. Singapura. Oke, Pak. Baik ya, Pak ya. Oke.
Oke. Ya, terima kasih banyak ya.
Prof Lim, terima kasih banyak.
Ya, terima kasih untuk kesempatannya.
Terima kasih, Prof Lim.
Prof Lim, terima kasih banyak, Prof Lim.
Oke.
Prof Chris, terima kasih. Sekaligus selamat ulang tahun buat Prof Chris.
Ya, terima kasih untuk teman-teman yang
juga.
memberi ucapan selamat ulang tahun. Tuhan memberkati. Prof Lim, terima kasih mengisi acara untuk acara ulang tahun saya. Ulang tahun kelahiran saya.
Ulang tahun Prof Chris.
Ya. Usia 70 tahun.
Ya,
masih still young, Prof.
70.
Masih. Ya.
Semoga makin sayang sama Bu Rita.
Ya, ini ulang tahun pernikahan kami.
Selamat Bu Rita, Prof Chris.
Ya. Jadi ini ulang tahun pernikahan kami, juga ulang tahun kelulusan kami.
Oke,
ya.
Hajatannya kapan,
Oke.
Prof?
Kenapa?
Ada itu nggak pesta-pesta hajatan?
Ada pesta tapi enggak.
Ada biasa
dengan
saja.
ulang tahun cucu saya, Prof Chris.
Ya, ya.
Cucu saya yang di Amerika.
Di Amerika ya.
Oke. Di Pittsburgh.
Wah, bisa join nih.
Iya, bisa join online nanti.
Ya. Kalau gitu, cucu nama diganti. Tambah nama Timotius gitu, karena tanggal lahirnya sama. Oke. Ya, terima kasih Prof. Terima kasih teman-teman semua. Tuhan memberkati. Ya.
Sekali lagi, selamat ulang tahun.
Foto, Pak. Foto, Prof.
Sudah foto toh?
Ya, ada foto.
Sudah.
Foto session before we leaving each other.
Ya.
Oke. Bye-bye.
Bye. Thank you very much, UKRIDA and Prof Lim and others.
Sama-sama.
Terima kasih, Prof.